So the first presentation is by Roxanne Jeffers, and her advisor is Chris Grant. For those of you attending the participation, if you're participating, I would like you to use the hand feature. All questions will be, we'll wait for the end of the presentation for questions. So at that point, if you can put up your hand using the hand feature, I will use that and call on you. Okay, Roxanne, go ahead and start.
Okay, so hi guys. Today I'll be presenting my research, which is on doing a comparative analysis of antibiotic levels in farmed rays and wild brook and brown trout. So I'll first start by giving some background as it relates to antibiotics. So an antibiotic is a chemotherapeutic agent that kills or inhibits the growth of microorganisms. And antibiotics are widely used in human as well as veterinary medicine. And in the agriculture sector, antibiotics are primarily used to promote the growth of livestock and prevent diseases. So, okay, so right now I have a question for you guys, and you can answer in the chat. So according to the FDA, more than 20 million pounds of medically important antibiotic drugs were sold for use on livestock farms. Antibiotics used in agriculture represent what percentage of all antibiotics sold? So I'll give you guys a few seconds just to answer that. Okay, so antibiotics used in livestock actually represent 80% of all antibiotics sold. So it's quite a lot. So currently the fastest growing area of the agriculture industry is aquaculture. And in the aquaculture sector, fish are in hatcheries and they're in very confined areas, and this leads to the spread of diseases. And due to this, hatchery suppliers give the fish large amounts of antibiotics in order to mitigate these diseases. And these systems produce a large volume of wastewater that is deposited back into our streams. And annually, approximately 3 million hatchery-raised trout are stocked in streams across Pennsylvania. So the widespread use of antibiotics has led to major public concern because we know that the overuse of antibiotics leads to antimicrobial resistance. And this is very dangerous because antibiotic resistance can lead to prolonged hospital stays, higher medical costs, and also increased mortality. And as we speak today, new resistance mechanisms are emerging and spreading globally. And there's a growing list of infections such as pneumonia, gonorrhea, blood poisoning, and foodborne diseases that are now becoming untreatable due to antimicrobial resistance. So due to the growing concern of antimicrobial resistance in the healthcare industry, this has led to further concern as it relates to the agriculture industry, namely aquaculture. And currently, the information about the persistence of antibiotics in wild fish is still limited, but we know that the continuous presence of antibiotics, even at low concentrations, can lead to the development of resistant strains of bacteria. And this has direct implications for not only the fish, but us. So this is another quick question. So in the United States alone, how many people get an antibiotic resistant infection per year? Oops, sorry. Sorry, you guys saw the answer. Did you guys see it?
No.
Oh, okay, I thought so. Okay, so it is actually 2 million. So 2 million people get antibiotic resistant infection per year, only in the US. So the objective of our study was to analyze and compare antibiotic levels of farmed rays versus wild trout. We utilize oxytetracycline as our marker antibiotic because it's one of the most commonly used antibiotics in the aquaculture industry. So the streams that we looked at were stocked in April and we sampled in July. Due to previous research suggesting that antibiotics may persist in fish, we hypothesized that we would find detectable levels of oxytetracycline in our samples. And our overarching hypothesis was that we would have a higher concentration of oxytetracycline in farmed rays than wild trout. So we obtained our samples by electrofishing. So we obtained our wild fish from 3 unstocked streams and our farmed raised trout from 2 stocked streams. And we dissected the fish and removed the liver, kidney, and muscle tissue. So we looked at the muscle tissue because it's quite known that contaminants bioaccumulate in the muscle tissue, and we looked at the liver and the kidney because those are the main excretory organs, and we utilized an analyzer in order to analyze oxytetracycline concentrations of our samples, and statistical analysis was done using Minitab 19. So before I jump into my results, I would just like to remind you guys of my hypothesis, which was that we would have higher levels of oxytetracycline in farmed rays than wild trout. So I would just like you guys to guess in the chat and let me know whether or not you thought my hypothesis was correct. Oh, sorry. So you can let me, sorry, I didn't explain it properly. So you can let me know which one is farmed rays and which one is wild trout. So whether A is farmed rays or B is farmed rays or wild. Okay. So this is our result. So this is the same box plot comparing oxytetracycline levels in the kidney of farmed rays versus wild trout. So we had a p-value less than 0.05. So this indicates that there was a significant difference. However, we saw a higher level of oxytetracycline concentration in wild trout versus farmed rays. For the liver, we also saw the similar trend of wild trout having higher levels, but the p-value was not significant. And for the muscle tissue, there was also the similar trend, but the p-value was also not significant in this case. Due to obtaining the result that wild trout had higher levels of oxytetracycline, we decided to do a by species analysis, and the same trend existed there. So overall, our results indicated that there was a significant difference in oxytetracycline in the kidney, with wild trout having higher levels of oxytetracycline. So questions still remain as to why this may be, but some reasons that we have, so it can be grouped into two categories, so environmental factors and also physiological factors. It is possible that the wild trout streams are being contaminated by other sources. And for physiological factors, it is possible that because the farm-raised trout are frequently exposed to antibiotics, that they are becoming more efficient at dispelling it from their system. The next step in this project would be to assess if this same trend exists under a larger sample size. So all of our oxytetracycline levels for the kidney, liver, and muscle tissue were below the FDA guidelines. However, it should be noted that the FDA has a pretty broad category as it relates to oxytetracycline concentration limits. So the limit is the same for the liver of the cattle or a fish. So, you know, cattle is much bigger than a fish, so it's very broad. And there has been previous research suggesting that the oxytetracycline has adverse effects on trout when they're exposed to it during their juvenile stages. So there is research that suggests that oxytetracycline can lead to spinal deformities in trout, and this poses issues for the long-term survivorship of wild trout. And even though our levels are low, we know that low levels of antibiotic can lead to resistant strains of bacteria. And for our future work, we would like to utilize PCR to compare antibiotic resistant genes between farmed raised and wild trout and see if a difference exists there. So I'd like to thank Dr. Mansberger for all of her assistance in the initial stages of the project and also all throughout my project, and then Casey Kaufman for her assistance with data analysis and fish collection. And then I'd like to thank Anik, Ben, and Seth for fish collection as well. And I'd like to thank Ian Schutz from Bioform for his assistance with ELISA. Yep, and I'm open to any questions or comments.
Great. Roxy, great job. Can you stop sharing your screen?
Oh, yes.
That would be helpful. So for all of you, if you have questions, please use the hand feature to just raise your hand. If I don't see anyone, I can start. It's a couple of screens, so understand it might be a little bit tough for me. Great job. I thought it was really cool that you had an interactive feature in your talk. My first question is, how did you identify hatchery versus wild caught when you were catching them in the stream?
So we went to known stocked streams, so streams that are known to be stocked. And that's how we knew that they were stocked. And for the wild trout streams, we went to Class A native streams. So that's how we knew that those were wild trout.
So the streams that were stocked did not have wild trout in them?
No, there were stock streams, yep.
Wow. Other questions, everyone? Don't be shy. I guess I can ask, okay, so there's a question here. Do you know whether the fishery that stocked the streams you sampled used antibiotics for testing? Do you know if that's the case?
Sampled and used.
So do you know that they actually used it? Did you have any interaction with the hatcheries at all?
So because oxytetracycline is one of the most commonly used antibiotics, that's why we utilize an ELISA specific for oxytetracycline. So we did not have direct interaction with the hatcheries, but after testing, we saw that we did have levels of oxytetracycline within the stock species.
Since nobody else seems to be asking questions, actually Jeremy has a question. Go ahead, Jeremy, ask your questions.
Good. So first of all, great job again, Roxy. I have two questions, if that's all right. So kind of piggybacking off of a previous one, have you thought about possibly using ELISA to test for concentrations of antibiotics?
Of other antibiotics?
Yeah.
Yes, yes. So the reason why we made it specific to oxytocin is because that was the most common one. But there's previous research, not with trout, but they look at various antibiotics. So like with more funding, we would definitely like to look at like a wide variety of different antibiotics. So yeah.
I was just thinking that maybe that's one reason why you saw the results that you did. But along with that too, have you thought about trying to do a control?
I would have to stop the question because
Okay.
we've run out of time.
Sorry.
Thank you. Great job. We will move on to the next presentation. Quinn Girasek, and her advisor is Vincent Buonacorrsi. Quinn, if you can share your screen, that would be helpful. Great. Go ahead and start.
Hello, I'm Quinn Girasek, and today I will talk about the phylogenetic classification of North American ratsnakes in central Pennsylvania. I just wanted to say Dr. Matter collected the snakes. Brianna, who graduated last year, carried out the DNA isolation and PCR when she was doing independent research here. And as part of BI 489 independent research, I performed the analyses. So just for some context, rat snakes are important because they are opportunistic predators and they keep the rodent and bird populations in check. So why are there different subgroups of snakes found in different locations? This could be due to different factors such as geographic barriers, interspecific competition, and climate and habitat differences. So before 2000, the North American ratsnake and the subspecies were taxonomically identified, classified under Elaphia obsoleta. So this research that we did extends on Burbrink's work, which found that there were 3 different clades or subspecies along the East Coast and the Midwest. The Western clade was the Pantherophis obsoletus, right here. Right here. The central clade was the Pantherophis spiloides, and then the eastern clade was the Pantherophis alleghaniensis. So these arrows, the map here shows the different sampling points that Burbrink used, and the arrows represent movement of the rat snakes after a glacier retreat that occurred like 6,000 to 18,000 years ago. Burbrink, as you can see in this map, didn't sample snakes from Pennsylvania, as well as snakes from the Appalachian Mountains, which brings us to the question of which clades are the snakes from Huntingdon County, Pennsylvania grouped as. Our hypothesis was snakes found in Huntingdon County, Pennsylvania were either part of the eastern or the central clades. To do this, our method starts with collecting living and dead rat snakes. The scales were obtained from the ventral side of the snake. So on this picture, you can see it right here. Then the snake scales were stored in 95% ethanol until the DNA extraction. After collecting the scales and storing them, then DDT was used to break down the scales. Afterwards, the DNEZ blood and tissue kit was used to extract the DNA. Then exosapit was used to purify the products. And for the PCR primers, we used cytochrome B from Burbrink. Singer sequencing was used to amplify the cytochrome B. And then after that, the products were then sent to the Penn State Genome Core Facility. Now for the analysis. The DNA subway web application was used to trim and pair sequences. And then the Mega version 10 was used to determine the best fit model of sequence evolution, which basically means the program looked through the data and used parameters such as the rates for different nucleotides. And from that, it presented a compiled list of models and parameters. Using that information, I made a tree using the maximum likelihood method, which is a method that produces a tree with the highest likelihood of accuracy given the particular data set. So in order to create the tree, select genomic DNA from Burbrink was imported to the program from NCBI's GenBank. And to create the most accurate model, a test of phylogeny was used using the bootstrap method with 1,000 replicates was carried out. So the bootstrap method indicates how well supported different branches are by resampling sites randomly. In the end, you get a phylogenetic tree that is based on resampled data and numbers that indicates percentages for how many times out of 1,000 each branch shows up in the same position. So basically, it's a support index. This table here, so for the results, this table here shows the PCR and the tree and some data from the tree. So we started off with 40 snakes. Oh, well, we collected 40 snakes. 13 were sequenced. The PCR yield was 1,116 base pairs long, and there were 1,010 sites after trimming the sequence, and 891 of which were conserved. This means that so the conserved sites are the parts of the sequences that are similar when comparing the different sequences. The variable sites are the opposite of that, and there were 818 of those. A parsimony informative site is a site that has more than one type of nucleotide, and at least 2 of those must occur more than twice. So there were 99 of those sites. Here is the phylogenetic tree that we constructed. For those of you who are not familiar with phylogenetic trees, they're grouped by genetic similarity, and this here would be a node, and this here would be a branch. So here you can see the western snakes make up, the snakes part of the western clade make up the outgroup, as is designated at node D. The individuals from the East Coast are grouped right here, and they make up the eastern clade. Node C shows where the eastern clade and the central clade split. At node B, you can see that the snake from Mississippi is separated from Pennsylvania, from the snakes that were found in Pennsylvania, Ohio, and West Virginia. And this may be the case due to Ohio and West Virginia being closer to Pennsylvania rather than Mississippi. For individual 122 here, you can see how it's sort of separated from the others found in Pennsylvania. And this could be the case because there were gaps in missing data from this individual, which influenced how the clustering looks. So while Burbrink sampled one snake from each county, we sampled 13 because we weren't sure if there was going to be any clade, and then having more individuals means that there are more frequencies if there were different types present in the county. So rat snakes found in Huntingdon, Pennsylvania Ratsnakes found in Huntingdon County, Pennsylvania are most closely related to the snakes that are part of the central clade as described by Burbrink. Huntingdon County is right here, located in the Appalachian Mountains. As I mentioned earlier, Burbrink didn't sample snakes from the Appalachian Mountains, but a different study by Matthews and colleagues found that snakes on the Appalachian Mountains were part of the eastern clade. So, but the thing is, they looked at the section of the Appalachian Mountains that was in North Carolina. Based on this information, it seems that there may be hybrid zones. The evidence to support our speculation of them hybridizing is based on Gibbs and colleagues. They studied snakes in Ontario, Canada. This map right here shows, the bottom part of the map shows where Burbrink sampled and Burbrink's results. And then the part up here shows Gibbs's study areas. So they conducted their study by using phylogenetic and hybrid zone analyses, and they also used mitochondrial DNA. They found that the snakes from the eastern clade and western clade are either hybridizing or there may be new species. The study sites in Canada were all like a bit west of the Appalachian Mountains, suggesting that the Pantherophis alleghaniensis snakes that were found to the east of the mountain range were somehow able to move across. So our study did have some limitations. We used mitochondrial DNA, not nuclear DNA. While mitochondrial DNA is good for phylogenetic studies, there are some disadvantages. So this type of DNA is only inherited through the mother, and it doesn't detect hybridization that well. So if a hybrid zone was found, then we could look at nuclear markers as well to see what in particular is going on. So further research can determine if there's a range in Pennsylvania or maybe just along the Appalachian Mountains where two clades of snakes were hybridizing. Here are my references, and I would like to acknowledge the Juniata College Biology Department for making this all possible. Thank you. Do you guys have any questions?
Thank you, Quinn. I'm hoping that, Quinn, if you can stop sharing your screen, anybody who has questions, if you can either use the hand feature or just the chat feature, that would be helpful. And Quinn, if you get up,
the little,
it's right at the top. You can just do, yeah,
okay.
Thank you. So if you can put up your hand or, okay, I see a clap for an excellent presentation. As you have a couple of, and this was great. I do have a question while you're waiting for others to sign up. I couldn't really understand why you chose to sample just 13 of the 40 snakes that you had. I would have thought that, you know, if you'd done a few more, you would have got more information. So.
Yeah, so I'm not too clear on that part because I wasn't really involved in that section of the research. Maybe
Fair
Dr.
enough.
Matter could explain, but.
There is another question from Dr. Powell, and he said, why are the mountains a barrier for gene flow given that black snakes can live in that habitat?
So just to answer your question, Dr. Matter just said for cost reasons. Then
Okay, fair enough.
for the other questions, we're not sure if mountains actually are a barrier, but due to it just not being sampled previously, it was like an opportunity or that could be studied.
We have another question earlier, which is, how long does it take to collect the snakes?
That one I'm not sure of. Dr. Matter might be able to answer that.
Yes, we collected these samples over a couple of years. To get the scales and so forth from the snakes is not a big deal. It takes all of about 10 minutes to trim belly scales on a snake, so that was not but it did take us a bit of time because it was totally opportunistic in terms of, cruising around and finding snakes dead on the road.
Professor Keeney has a question about asking about whether other genes have been used to define clades other than what you used, the cyto B.
Well, we used cytochrome B because that's what the other paper by Burbrink used, and just so we could compare the results easier. That's why we used it. I'm not sure if other genes have been used, but maybe like with other organisms.
Okay. If there are no more questions, again, excellent presentation. We will wait for half a minute before we move on to the next presentation. And the next presentation is by Brianna Geiner and Joseph Franckiewicz, and the faculty advisor is Dr. Chuck Yohn. And if you can wait for 10 seconds, that would be helpful so we can let everybody in. Okay, if you can, Joe, if you can share your screen and start, that would be great.
Of course.
Thank you.
Our study was looking at oak regeneration within 2 different aged plots after a shelterwood cut in a central Pennsylvania hardwood forest. My name is Joe Franckiewicz. And also with us is Bree Geiner. I'm not sure if Bree is able to hear us. Are you good, Bree? No audio.
Bree, turn on your audio. Joe, can you continue while we see if...
Of course, yeah, I'll just take over. So we were looking at oaks, which are a very important timber species. There's a couple reasons why oaks are a desirable timber species. For instance, they are known for being a good food supply for wildlife. They provide hard mast, leaves, twigs, shoots, and acorns, which are very important for deer and squirrels and such. They're also a very important shelter tree as far as providing shelter for wildlife. They maintain watershed integrity with their root systems. They are aesthetically pleasing to look at. But the one reason that we chose oaks is because they are a very important timber species. They produce very high quality grade lumber, which makes them a very desirable species to grow here in the Northwoods. However, one issue that's been happening with them is that they have been on the decline.
Okay, can everybody hear me now?
Yep.
Yes.
Okay, sorry for that. I'll jump right in. Okay, so as Joe just finished is that we do have issues regenerating oaks. Some of these issues that can impede regeneration are the loss of fire, low light levels, the competition from other growth, and deer browse. Oaks are very sensitive to light, and it greatly affects its success if there's other species blocking this or creating shade. Deer browse is also a very heavy tormentor on the landscape because there's so little for them to eat. All the new growth that's available, they just kind of decimate the likeliness of success and eat everything.
So with these issues in mind, how do we successfully regenerate our oaks? So there's one method that's been gaining a lot of popularity in regenerating oak stands, and that is called the Shelterwood Method. It is a 3-phased cut, which consists of your preparatory cut, your establishment cut, and your removal cut. So that first phase is your preparatory cut. This is the first thing you do when you go into the stand. If there's any thick vegetation, you remove it. If you were to do an herbicide treatment, you would do it during this phase. If you were to do a prescribed burn, you would also do that during this phase. And the purpose of the preparatory cut is to scar or disturb the soil, which helps prepare the seed bank for germination. And when I say seed bank, I'm referring to seeds that have already been laid down by nearby trees, things that are waiting dormant in the soil, waiting to sprout. Your second cut is your establishment cut. This is going to be the largest cut that you do during this phase. This is when you go in and you provide space for seedlings to grow. Typically, you're going to remove 40 to 60% of your canopy, so that's the overstory, and you want to leave behind the strongest, best quality trees, and those are going to be what you call your shelter trees, hence the name Shelterwood. And the reason why you leave these behind is to provide cover and a little bit of shade for your seedlings, as well as shelter for wildlife and such. Your third and final cut is done typically 10 to 15 years after your establishment cut, and that is your removal cut. So during the removal cut, the purpose is to completely remove that canopy that you left behind during the establishment cut. So as I mentioned earlier, you're probably going to have 40 to 60% canopy cover left over after the establishment cut. During the removal cut, you get rid of that remainder. And this is to allow a surge of light to reach the forest floor and allow your seedlings to grow rapidly.
So our research question is, how effective is using this Shelterwood cut method in regenerating oaks, particularly within our native central Pennsylvania hardwood forests? This method looks great on paper and works in other locations, but how will it fare within our woods? So our study area. Our study area was a timber stand near Raystown Lake called Putz Camp, next to or adjacent to a Boy Scout camp. We had the delight of staying at Raystown Field Station for our forestry semester in the fall, so it was very easy to conduct our research close by. This is an aerial view of Putz Camp. It is outlined in white. Within the timber sale, there was an earlier cut section done in 2018, and that was broken up by a deer fenced area. We did not include this in our research. If you look at this aerial view, off to the right is stand 1, and off to the left is stand 2. So looking more closely into our stands, stand 1 was the largest size at 60.42 acres. The date it was cut was August to October in 2019, and the species within it consisted of oaks and hickory, which is very consistent with our central hardwood forest species makeup. Looking at stand 2, it came in about 9 acres. Again, it was slimmed down due to this deer enclosure that we did not include. It was cut from September to November in 2018, and it had very similar species growth, also being oak and hickory.
We used a random sampling technique to find 10 random points in each stand that Bree talked about, leaving us with 20 random points total. At each point, we laid down a collapsible meter square, which you could see in the photo on the right. And we recorded any species that we noticed within that meter square. If it was below 25 centimeters in height, we did not include it in our study. This is because a similar study we looked at determined that once a seedling reaches 20 centimeters in height, it has a greater chance of survival. So we went with 25 to be on the safe side. We also tallied the amount of species that we found in each point. And if we noticed any herbaceous plants like blackberries or raspberries or stiltgrass, we did not include those in our study. We were only focused on woody plants. As far as our statistical analysis went, we used 2 different tests. Our first one was a Mann-Whitney U test, which compared the median densities of oaks per acre between stands. This left us with an estimated stem per acre count of either stand. Our second test was a Pearson correlation test, and we used this to compare the stem densities, so that stem per acre count of oaks, and compare that to a stem per acre count of tulip poplar, which is another type of tree that was found on that stand, a species that our forester Glenn Warner was trying to propagate. So we were trying to see if the presence of tulip poplar affects the presence of oak species.
So looking at our results, within both sites, there was a wide range of species that we recorded, and we kind of put them into 2 categories of target species and non-target species. The non-target species that we recorded were first successional species such as dogwood, redbud, fire cherry, black gum, and black locust, and they totaled around 5%. There was also black birch, sugar maple, white ash, red maple, and sassafras. These were other species that totaled around 19%, for a total of 29% in that non-target species category. In our target species, also known as desirable species, were our red, white oaks and our tulip poplar, which were the bulk that we recorded at 71%. So the median density of our oak seedlings was lower in stand 2 versus stand 1. And there was a medium negative correlation that existed between the oaks and tulip poplars in both stand 1 and stand 2.
We compared our findings to the findings of similar studies to see what other researchers were deeming as successful oak regeneration. When looking at similar studies, researchers were determining that anywhere from 2,000 to 7,000 stems per acre was deemed healthy regeneration. When we compared this to our own findings, we found that both stands fell within this reported literature range. Our stand 1 cut, or our 2019 cut, had 5,600 stems per acre, and our stand 2 cut, or our 2018 cut, was at 2,800, which is on the lower end of that spectrum. So we could see that as time goes on, we are losing oaks, but it's still within that reported healthy range. We did the same for tulip poplar and found that other researchers were ranging anywhere from 200 to 2,500 stems per acre. When we compared this to our own findings, we found that in our 2019 cut, we had 1,600 stems per acre, which is within that healthy range. However, when we looked at our 2018 cut, we found 5,600 stems per acre, which is above that healthy reported literature range. And it does show us that as time goes on, we're actually increasing in the amount of tulip poplar that we're finding. So as far as future management implications go for the future of Raystown Lake, we have a few suggestions. First and foremost, we'd like to suggest that if timber management is done at Raystown Lake, that the Army Corps of Engineers continues to use the Shelterwood method, as it has proven to increase the amount of oaks or other desirable species on that site. We also recommend that if they continue to do Shelterwood method, that they also continue to use herbicide treatments during their preparatory cut, as we did find a lot of invasive species on the site, notably Japanese stiltgrass. We've discussed the possibility of controlled burns on the site. However, this would have to be weather permitting. We have found throughout many studies that fire is a very valuable tool with regenerating oaks, but it has to be very weather dependent. Our final recommendation is that if we continue to do shelterwood for oaks, that we do not use tulip poplar as shelter trees. We suspect that tulip poplar is a strong competitor of oaks, and if we're trying to regenerate them, it's not beneficial to have tulip poplar alongside them.
So in conclusion, our research supports that our Shelterwood method is very successful, and it's a very successful way in general to regenerate oaks, and in this case, in this study, tulip poplars.
We'd like to thank our forester, Glenn Werner, for providing us with any information we needed about the timber cut as well as the site. We'd also like to thank Chris Baumgartner for being one of our advisors on the project, as he provided us with access to our timber site, and he helped us when our van couldn't get into our study site. We'd also like to thank Dr. Chuck Yohn for providing us with any information we needed about Raystown Lake and its natural history. He also helped us with our statistical analyses and was our primary advisor on this project.
We'd also like to thank our research team that helped us collect all this research to begin with: Maya McDonald, Pam Cray, Max Mueller, and myself and Joe.
Okay, Joe, could you stop sharing your screen? Thank you very much. Excellent presentation. If anyone has questions, if you could open it up. We have about 2.5 minutes for questions, so go ahead. Again, I guess I'll have to start. The technique that you used, is it like, wait, sorry, Dr. Muth has a question. If we can start with him.
Yeah, sure. Nice job. Nice job, both of you, on this presentation. My question was about deer densities, I suppose. There was a deer exclosure adjacent to at least one of these, maybe both of the sites. but the data you have is for areas that that were not fenced, yet it looked like you had quite a bit of sufficient regeneration. I'm just wondering, it seems like it wouldn't have been exactly part of your study, and I assume you don't have data for those deer exclosure sites, but what's your sense there, since putting up those fences is extremely expensive and burdensome, if you have some insight as to whether things were going on differently in those adjacent sites, and just sort of what your thoughts are there.
So I can answer that. We did not include the deer exclosure in our study as per the request of Glenn Werner, who was our contact with the Army Corps. The reason why is because if we were to look inside the deer exclosure, it was visibly denser as far as stem counts went. He wasn't interested in looking at a site that he can already tell was a lot denser. We did find sufficient regeneration, but there absolutely was evidence of browsing, and that particular area is a DMAP zone, so we can tell that there's already an abnormally high amount of deer that are already there, but we just didn't include it.
Yes, as Joe said, there was also evidence of deer within the deer enclosure. So deer did find a way to get in, and they did definitely probably eat some of the regenerated growth. But
yeah, that's a classic problem.
So I'm sorry, we'll have to stop here. I know that there are a few more questions, but we've run out of time. So if you could stop sharing the screen, oh, you have. Stephanie, could you go ahead and share the screen? And so our next speaker is Stephanie Letourneau, and she will be talking about her work during her internship. Go ahead and start.
Yeah, can you guys see the presentation okay?
Yes, we can.
Perfect, okay. So again, hi, I am Stephanie Letourneau. I am a senior, and this is from my research this past summer, part of the Holling Scholarship with NOAA. A big shout out to Dr. Buonacorrsi. Not sure if he's still on the chat, but he is the professor that helped me get this scholarship, and I will be forever grateful. And I actually was able to work at the Chesapeake Bay National Estuarine Research Reserve on the campus of the Virginia Institute of Marine Science, which is his alma mater. So I have been continuing with this research, and that's why I decided to share it with you guys today. And I think it's very important for scientists to understand what the results were of my research. So I worked in the National Estuarine Research Reserve System, and if you aren't familiar, the acronym is NER, or NER, depending on what part of the country you're in. And there are 29 different coastal sites. So you can see I was at the coastal site with the star on it in Chesapeake Bay, Virginia. And what's really unique about having these NER sites is that they're able to do large-scale collaborative research. So they're able to look at different effects of different coastal projects in different parts of the world, in differing ecosystems, differing societies, etc. So this was a very unique system to be a part of and to be able to participate in research with. And part of my internship is I worked with our stewardship coordinator and our education coordinator to kind of be the link between the two of them and make sure that our stewardship team and all their research is being promoted. And one of those projects was called the Thin Layer Placement Project. And thin layer placement, if you look at the picture on the bottom left, that is a picture of large scale thin layer placement. It looks a little scary. That picture, they're throwing dredged material on top of the marsh. And the whole idea behind this is that they're going to build the marsh up to be able to withstand sea level rise. Many of us in this chat are environmental savvy, sea savvy, so we know with climate change, we're having sea level rise. And this is a huge problem for our marshes because our marshes are what kind of blocks our coastal sites from the energy of storms and waves, and it filters out nutrients, and it's a nursery habitat for many of the food we like to eat, the birds we like to see. So it has a lot of ecosystem services. So this experiment was for 2 years at 8 different near sites. I got to go to Goodwin Island, one of our sites. And what we were doing is we were putting sediment of different thicknesses and different compositions in these squares, if you will, and looking at over the years of how the marsh is building itself back up, what species are coming up, and can we make it more resilient using this strategy? So as part of my research and my internship, I got to go out with the stewardship team, and I was able to do a lot of cool things. That picture of me holding that muddy tube is a soil core. So I was taking a core of soil to pick up all the roots in the soil so that I could do a bunch of analyses to see what's in the soil. The other picture is called a surface elevation table. It's all about making sure we have a datum. So making sure that we are looking at things with reference to what's actually going on rather than our data being skewed by, you know, climate change or subsidence, which is the sinking of the land. And then after I did all this data collection, my job then with the education coordinator was to develop lesson plans and activities to be used. And I was doing that, and I also was trying to make another dream come true for them. And they said, well, you know how to use GIS. Can you figure out how to make it build a story map that can be used with this as well? And what's really interesting about story map, and I'm actually going to take you to my story map, if I can manage this, we'll see. Can you guys see my story map?
Yes.
Perfect. Okay. So Story Map is one of Esri's many platforms that they have for you to be able to share your research and share your maps with other people. I'm trying to get to one here. So this is an interactive map that is using shapefiles from GIS.
I'm sorry to interrupt. Apparently, this is not the story map. That's not what we're seeing.
Nope, that's okay.
Okay.
I will get you to it.
You have to change the share screen.
Are we there now?
Okay, thank you.
Perfect. Okay, so take everything I just said and put it onto this slide. And so this is a map showing the different vegetation types of Goodwin Island. So this helps students be able to understand what this kind of looks like. And I took my whole lesson plan and put it into this interactive format for students to be able to interact, learn about thin layer placement experiment, learn about the nears, but also participate in an activity. So I made the lesson plan, I made the story map, and they can see where the different sites are. Really fun. Okay, I'm going to take you guys back to my PowerPoint. So I made the lesson plan, I made the story map, and I now have to look at, okay, what is the effects of teaching using my lesson plan versus just the story map? One of the things I had to do with my internship was actually do a research project. So I went ahead and looked at what the perspectives of students and teachers of doing this kind of research and learning using the story map versus traditional learning. And you might be wondering what digital learning is if you're not in the education field. Well, digital learning is using any kind of technology to be able to enhance learning in the classroom. A lot of us are doing emergency remote online learning right now. That is different than what digital learning is. I'm not talking about an online class. I'm talking about using technology in the classroom, different than what we used to use, such as graphing calculators and PowerPoints. This is much more now. And so the way to do this was a completely new methodology for me. I did a phenomenological study, which included semi-structured interviews. I was working with students and teachers over the summer, which are hard to access. So my first group was with the teachers. I helped with a Teachers on the Estuary workshop, where I interviewed the teachers and asked them about what their opinions of technology in the classroom was, how they implemented technology in the classroom, what issues they were having, pros and cons. And during this time, I was able to really understand, you know, where we have flaws in our system of using technology. And these were all science instructors. So it was very focused on what I was trying to look at. I also looked at students in a summer camp, and I did the whole lesson plan and the story map with the students, and then interviewed them and asked them, how did this make you feel? And these were all students who were incoming 9th graders and incoming, we had one college freshman. And so it was hard actually to do these interviews with them because I had to really push them to give me information. High school students don't like to talk too much about how they feel. So I got all this information, all these opinions. I had to go through and code these interviews, and I'm still compiling what that information looks like, what the themes out of those interviews are. But during this, I also finished my lesson plan and my story map and sent them out for evaluation. So my teachers actually ranked my lesson plan, and they were able to tell me what they thought of it. So this is some results of the interviews. Lots of advantages, lots of disadvantages, and this is all about technology in the classroom. You can see distractions and misuse has a star. That's because that was the #1 issue that my teachers mentioned. And it kind of comes back to one of the things in the advantages is that students are now growing up with an iPhone in their hand, and we need to help aid them in becoming digital citizens to learn how to use this technology to be researchers in this social media technology world. And once we do that, maybe, just maybe, those distractions and misuses are going to go down. So these are the results of my evaluations from my teachers. These aren't statistically significant for those of you who are stat savvy, but as you can see, I only had 9 evaluations from my teachers out of the 14 I interviewed. Teachers are busy over the summer doing vacation. And my story map ranked higher than my traditional lesson plan, which was interesting because there was nothing really new about it other than the format it was in and the interactive tools. So for my students, I have just 2 quotes from them to really talk about what they actually felt, because they all excelled. They all did really great. I did a pre-test and a post-test after my pilot with the students, and they all got 100% in the end, and they got 0% in the beginning. So they obviously learned something, and they all really enjoyed it. And there was a bunch of different reasons for that. And so, you know, it's auditory learning, it's interactive information. And if you have more questions, I'll be happy to help you, let you know what students think. But in the end, I kind of have concluded that we kind of need to start incorporating, especially in science classrooms, this blended learning. Students really need to be digital citizens now. It's really hard for us to have technology in the classroom without actually teaching them how to use it and what it's useful for, especially in STEM, where we're doing all of this analysis on our laptops, on our computers, we're doing modeling. Students need to learn from the bottom when they're in elementary school what that looks like. And our education should implement more resources, but with that, you know, we need to provide as scientists more resources for these teachers. They can't always just come up with things on their own. And the plus side of that is that our teachers can get real world information, which students found that they preferred a lot more. They don't want to just add up random numbers and do random analysis on fake data. They prefer having that real world connection. For example, my students were looking at data that was completely real. This was from the exact study. So it makes a lot more sense to have that in our classrooms. And I would like to thank the Holling Scholarship Program and my amazing mentors, Sarah Nuss and Scott Lerberg, and the Juniata Environmental Science Department. I got funding from the Environmental Fellows, which allowed me to go to a bunch of conferences this year and actually get a lot of people's perspectives on my research and kind of help me figure out what I'm doing with it and with my career and how to go forward. And if you're interested in visiting the story map, you can scan the QR code. And I'll also share the links in the chat. Uma, you're muted.
I realized. Presentation. So unfortunately, we have run out of time. So this was an amazing session. All of you did a great job. Stephanie, again, I tried to show you that you were running out of time, but I understand. It's so if you have more questions for any of the speakers, please email them with questions. And Stephanie has put a link to her, I'm guessing it's the storyboard, is that correct?
Yeah, story map.
Story map on the chat. Great job, everyone. Thank you.